Continuing our discussion on the role of metabolism in cancer, specifically how the alteration in metabolism can not only lead to cancer but promotes its progression, one of the most important process which comes whenever talking about the metabolism and cancer is the glycolysis. You know about glycolysis is one of the very major metabolic pathway, one of the metabolic pathway which is associated with many other metabolic pathways also. It's central in the glucose metabolism. It's fundamental in both normal and malignant cells. What you know, it starts with the glucose as the major molecule and or reactant, and at the end you get the pyruvate. This you can see we started with glucose. It's not new. We have been dealing this topic again and again, so it must not be new for you. So glycolysis effect in cancer, I mean how glycolysis is altered, or how the cancer cell depends so much on the glycolysis. It is also known as the Warburg effect, which I have already mentioned. Actually, Warburg was the first scientist who in 1923, 100 years ago, had already showed that somehow the cancer cells, the malignant cells depend more upon glycolysis as compared to Krebs cycle. This also must let you know, and that it's not all the time that you should have the best of technologies in your hand to come across some finding. If you are curious enough and if you are persistent enough, you can still manage to come across some very important, some very actually pivotal findings. And this finding, which showed the role of metabolism in cancer, which was somehow discovered some 100 years ago, is one of the hottest topic when we are talking about cancer in the 21st century also. This shows the importance of having passion and motivation in learning or in asking questions. It's very much implicated in understanding. Not only it tells you how we can deal with cancer, it can also help us to understand the different aspects of cancer. You can also ask the question here, and I asked you actually the question, why, what benefit the cancer cells have for that they for requiring or for depending more and more upon glycolysis as compared to Krebs cycle, for instance. So क्या फायदा हो सकता है? What could be the best benefit they can get out of it? Now, Warburg effect, what actually happens with the Warburg effect, that there is your cells start depending more and more on glycolysis. And how you know it? That there is much more transportation of glucose through the glucose transporters in the cell, which can be then used for the process of glycolysis. For example, glucose transport 1 in the lung cell cancer, like lung adenocarcinoma, a very major lung cell and very, I must say, a serious lung cell disease or lung cancer disease. In this one also, the glucose transporters, they are overexpressed and their activity is enhanced. And if you have highly expressed glucose transporters in this cancer, it will mean that prognosis of survival is low. There have been many functional studies which have been done on glucose transport 1, and for instance, if we knock down the glucose transporter once, it will lead to the decrease in the cancer progression or in the proliferation of the cells which leads to cancer. And also, it also reduces the metastasis by reducing the migration of the cells from one tissue to the other. Then, uh, as I already mentioned, that glycolysis is increased in these cells. And this increase of glycolysis, of course, depends on the enhanced influx of different molecules which are used for enhancing the process of glycolysis. First of all, I already mentioned that glucose transporters are involved in it, and also then they in turn lead to enhance the uptake of the glucose in the cancer cells. And this is the technique which is then which this actually enhance uptake of the glucose by the cancer cell. This is used when we are dealing in the diagnostics section in the for instance in the PET scanning. What we happen in the PET scanning, we may make use of this principle that because glucose uptake is enhanced, we used a radio-labeled glucose isomer, and that radio-labeled glucose analog, whatever you can say, this is then this is also taken by your cells, cancer cells, because the cancer cells have enhanced now their ability to get the or to uptake the glucose. Now they will uptake more and more radio-labeled glucose, and in this way, this radio-labeled glucose will be used to detect the cancer through the PET scanning. Here you can see how you can also make use of the natural systems for the diagnostic purposes. Then there are key regulatory enzymes, which is most important of this is hexokinases, which are of course involved in the phosphorylation of glucose. Then also another hallmark is that you know, in the aerobic respiration, what happened that pyruvate is converted into Krebs cycle. But because these cells depend very much on the glycolysis, वो क्या करेंगे फिर? They will be depending more and more anaerobic respiration, in which they will be producing more and more lactate. So production of more and more lactate is also one of the feature of the cancer cells. And we know that cancer is a genetic disease. Genetic in the sense there are certain genes which become mutated. Most important of these, of course, are the oncogenes, which once mutated, they leads to the proliferation or overproliferation of the cancer cells. Two of the most important of these are HIF1 and c-MYC. Both of these lead to the more and more dependence upon of the cancer cell upon the glycolysis, more and more uptake of the glucose, and then there is more and more glycolytic activity. Then there is also a tumor suppressor genes which are implicated in this in the role of metabolism in cancer, because when there is more and more glycolysis in the cancer cell, one of the reasons is that the tumor suppressor genes are suppressed. And this has been shown, or this has been seen when we saw that when we deactivate the p53, it promotes the glycolysis dependency of the cancer cells. So in this way, the loss of p53 leads to the promotion of cancer, we know, and one of the factor in this is the dependency of the cancer cells on the glycolysis, because loss of p53 leads to that dependency. So both the oncogenes, the overexpression of the oncogene and the suppression of tumor suppressor gene like p53, it leads to the more dependence on the glycolysis, and which ultimately leads to cancer promotion. Some of the mechanism through which HIF1A can leads to cancer because of its dependence of the cancer cells on the glycolysis is that it leads to the overexpression of the glucose transporters, also upregulation of the enzymes of the glycolysis like hexokinases, like fast fructose kinases, and like pyruvate kinases. It enhances the lactate dehydrogenase enzyme and MCT4 expression, and these also lead to the increase in the anaerobic respiration. You will get more and more lactate, one of the feature hallmark of the cancers. Then both c-Myc and HIF1A, because they are oncogenes, they collaborate with each other, which ultimately enhance the pathways, which leads to the pathways like PI3 kinase pathway, a very important pathway. This pathway leads to the promotion of cancer. So of course, when we are talking about cancer, when we're talking about the role of metabolism in cancer, then of course we have to talk about the therapeutic implications of this relationship. Then one simple way is that you then inhibit the glycolytic enzymes, which are on which the cancer cell depends so much, so heavily they depend. You can also transport the glucose transporters. You can, because lactate is more and more being produced in this, and it enhances the devastation caused by the cancer. You can also exploit the or target the lactate production. One way of dealing with treating the cancer is when we're talking about the enzymes which are involved in the glycolysis is, of course, you can use the hexokinase inhibitors like 2-DG, 2-deoxyglucose. It disrupts the glycolysis. Then you can also activate the pyruvate kinase, which then leads towards the Krebs cycle. So cell will depend more and more on the Krebs cycle as compared to the glycolysis. And you can also inhibit the glucose transporters and lactate dehydrogenase inhibitors, because more production of lactate is also one of the hallmark of cancers. Because of the more production of lactate, there is more acidic environment, which in turn promote the malignancies and metastasis. So you can neutralize the acidic environment also. And in this way, you can treat cancer both by using glycolysis inhibitors and also other conventional techniques of chemotherapy or X-rays or other type of radiations.